Good morning, everyone. Welcome to another edition of the RePo-Webinars, and an initiative from the RePo-SUDOE project for the promotion of scientific knowledge and dissemination of technology for drug repurposing in the SUDOE space. So first of all, thank you all of you for joining our webinar, and thank you to our speaker today, Joaquim Aguirre, for the availability for giving this presentation today. How it will be the structure of our webinar. It will follow this brief introduction that I will do. We will have the scientific presentation from Joaquim Aguirre. After that, some minutes for the discussion over the presentation, and finally closing of the webinar. All these within the total time of 1 hour. So our speaker today, it's Joaquim Aguirre Plans. Joaquim is a computational biologist at STALICLA, a clinical stage biopharmaceutical company pioneering neuroprecision medicine in developmental and psychiatric disorders. I guess Joaquim will also speak about this. Joaquim is a computational biologist at STALICLA, specializing in drug repurposing for neurodevelopmental disorders. Joaquim holds a PhD from the Universitat Pompeu Fabra, where he developed network medicine tools for disease research, pharmacology, and drug safety. He has also conducted postdoctoral research at the Barabási Lab on RNA-Seq sample size effects in network medicine analysis. So the presentation of Joaquim today has the title of Network Medicine Computational Tools for Drug Repurposing. And without further ado, please, Joaquim, the screen is yours. And thank you again for the opportunity to having you here at the RePo-Webinar.
Thanks to you for inviting me. Okay, so hello, my name is Joaquim Aguirre, and I'm a computational systems biologist at STALICLA, a pharmaceutical company that does research on neurodevelopmental disorders, applying personalized medicine. And today I will talk about network medicine computational tools for drug repurposing. So let's start by looking at how drugs are traditionally developed. The process is usually divided in three main phases: drug discovery in the lab, preclinical testing in animals, and clinical research in humans. But as you can see at the bottom, the first two big challenges of the drug discovery are very clear. It takes too much time and it costs too much money. But actually, there is a third major problem. It's called the lack of translation. And what does this mean? Well, it means that many drugs that seem to work well in the lab or in animal models fail when they reach human patients. Between preclinical research and clinical trials, there is what we call the valley of death, where promising discoveries simply don't make it across. This gap is one of the main reasons why drug discovery is so inefficient. So how can we improve this situation? Well, we need to find drugs that are effective, but also in a way that is fast and cheap. That means avoiding the traditional discovery pipelines and safety procedures whenever possible, and instead focus directly on the mechanism of action, understanding how the drug works and whether the mechanism can be used to treat the disease of interest. But the question is, how can we do this? Well, in order to find drugs spending less time and money, the idea is to repurpose existing drugs. Those that are already known to be safe and test whether they can be redirected to fight other diseases. And then, in order to find drugs that are more effective because they target the mechanism of action of the disease, we can use network medicine, which means to model the interactions in our cells as networks. So in today's presentation, I'll first explain what is network medicine, and then I'll show you 3 different network medicine tools that for me are very useful for drug repurposing. So I'll first try to cover a little bit the basics very quickly here. So proteins are the structural and functional units of cells. Structural because they form the scaffold of the cells, and functional because they carry most of the molecular processes of the cells. For example, the insulin is like the protein that regulates our blood sugar but still insulin cannot do this on its own. It needs to bind another protein, the insulin receptor, which sits on the surface of our cells. And only when the insulin interacts with its receptor, a signal is triggered that allows the glucose to enter the cells. So without this protein-protein interaction, the function simply cannot happen. And this happens with most of the cells, with most of the proteins in our cells. They tend to interact with other proteins, forming complexes to complete their function successfully, and therefore the study of these interactions is key if we want to really understand the molecular mechanisms of our cells. For this reason, a field called network medicine emerged several years ago with the objective to compile all the molecular interactions in our body and organize them as networks so that we can analyze them using network science algorithms and understand better the mechanism of the diseases and how to find new treatments that are personalized and target the mechanism of action of the disease. But how can we do this? This is the question. Well, the solution is to create a network that englobes all the protein-protein interactions in our organism. We call this network the interactome. And in this network, the nodes are the proteins, while the edges are the physical interactions between these proteins. But now that we have the interactome, how can we study and understand the mechanism of a disease? Well, many diseases are caused by genetic variants, which are modifications in the sequence of genes that are detrimental for the homeostasis of our molecular functions, which causes the disease. So these genetic variants are also called disease gene associations. And there are different types of disease gene associations, but among them, there are the ones that are originated by alterations in the coding regions of the DNA. And this means that if there is an alteration in these regions, it affects the protein that comes up from these genes. So if these genetic variants produce the loss of function of the protein encoded, this subsequently affects the disruption of the interactions of this protein, provoking a disease. These disease gene associations are characterized because their gene products are not randomly spread in the interactome. As it was described in 2011 by a review by Laszlo Barabási, these proteins are these proteins that are associated with a specific disease cluster together in specific regions of the interactome that we call disease modules. So identifying these disease modules, these clusters in the interactome, and analyzing them properly is the key if we want to understand how diseases work. Also, in 2016, Emre Guni and colleagues demonstrated that drugs are more likely to be effective when their targets lie close to a disease module, especially when they hit the proteins inside the module itself. So this is very positive because if we find the disease module and we find these targets that target this module, we already have a list of candidates that could be promising. And they even propose a formula that it's called network proximity to rank the drugs by their closeness to the disease module. And this formula, it's basically based on calculating the average shortest path between the genes of the disease module and the targets of the drug, and comparing this to a random distribution of distances. So this would be a summary of the main concepts behind network medicine. First, create and analyze the interactome, then identify the disease module, and finally, prioritize drug repurposing candidates by proximity. But now, how can we easily put these concepts into practice? So here I will present you 3 computational tools for drug repurposing using these network medicine concepts. And you see that I classify them according to how easy it is to use them, and also the level of detail that you can achieve by using them. And one thing that is clear is that the more detail that you can achieve by using these tools, the more complex that it also becomes to use it. For example, GUILDify, the first one is a web server, but NetMedPy and the disease module discovery pipeline are pipelines and packages which require you to already know a little bit of programming, which means that it's already much more complex to use them. So I will start with GUILDify, which is a web server that I helped to develop when I was doing my PhD here in Barcelona. So I will explain you the theory behind it, and then I will make a quick demo so that you can see that it works, or it should work at least. So GUILDify is a web server that allows to explore the mechanism of diseases, drugs, and then the relationships with them through the interactome. So let's start by how to find the disease module using GUILDify. The tricky part here is that finding a disease model is that we don't know all the disease gene associations. That means that the module isn't always as obvious or complete right from the start. So what GUILDify does is to take the known disease gene associations as seeds to run an algorithm to uncover the additional connected genes starting from these seeds. So put together, these genes form the disease module. So the main advantage of GUILDify is that it simplifies this process a lot. So basically what you have to do is to enter the GUILDify homepage and to type the name of the disease of interest, for example, asthma. You can also select the type of interactome that you want to use, because we have interactomes from 7 different species and 22 different human tissues. So you select them and you click to search. And then what it happens in the background in GUILDify is that when you click to search, the web server searches for genes associated to the disease of interest, to asthma, for example, in 3 different databases, DisGenet, OMIM, and UniProt. And then it redirects you to another page in which you can select which disease gene associations you want, and you can filter them manually or by database. If you have a database that you prefer, you can just filter it. You also have to select the algorithm that you want to use to identify the disease module. And once that you select it, you click to GUILDify. And then what happens first is that the server places the gene products of the selected disease gene associations in the interactome, and then applies a network diffusion algorithm, which basically what it does is to pass a message that starts from the seeds to the rest of the proteins in the interactome, and then it scores them. It gives a score that it's according to their closeness, but also to the topology that this area of the network has. So in the end, you end up with a lot of proteins and a score. And then what you have to do is to apply a threshold to select the top scoring proteins associated to the disease. And once that you do this, you will already have the disease module. And an example of threshold that we typically use is, for example, to select the top scoring 1% of proteins in the interactome. So this process can take some minutes, but after waiting a little bit, the web server redirects you to a results page that looks like this. And this page gives you the final ranking of proteins ordered by the score, and then a visualization tool that allows you to inspect the disease module and the associated drugs. It also provides you with the enriched functions and pathways of the genes of the disease module, which will facilitate a little bit the analysis of the module and give you an idea of if the module makes sense biologically or not. And then GUILDify also permits to explore, instead of a disease, like the perturbation that a drug can provoke in the network. And the process is very similar as before. So you go to the homepage, and instead of putting the disease name, you put a drug name, like erlotinib, for example, and click to search. And in this case, instead of searching in disease gene databases, it will search for targets associated to the drug in 4 different databases, which are DrugBank, Drug Central, DGIDB, and Campbell. And once that you select the targets of interest, again, it will compute the module, and then it will redirect you to the results page, in which it will show you the drug module, the proteins affected by the drug, and you can explore it. And again, it will also show you the enriched functions and pathways. And now, a very cool thing that you can do with GUILDify is that it also allows you to explore the relationships between 2 different results. For example, 2 diseases, or a disease and a drug. And this is very simple. What you have to do is to go to the homepage, where there is a section called overlap between 2 results. And this will allow you to introduce the job ID of 2 different results that you have computed before. For example, the ID of the disease and the ID of the drug, and then click to check overlap. And GUILDify instantly redirects you to a results page that shows you the genetic overlap between the modules of the 2 results introduced. And by using a Fisher's exact test, it also shows you the significance of this overlap. So how likely it is that this result is not random, that the overlap is not random, and that the two entities, the disease and the drug, or the disease and the disease, are really related. You also have the list of common proteins or genes, and then a visualization tool that also allows you to see what are these common, how are these common proteins related in the network? How are they connected? Another very cool thing is that apart from the genetic overlap, it also shows you the functional overlap, which is the overlap between the enriched functions between both results. And again, it also shows you the significance of this overlap and which are the common functions. And finally, it also calculates the enriched functions of the common genes. So you have everything here in the results page. And finally, we also have like another very cool feature, which is to search for drug repurposing candidates. So here you go to the homepage again, but now to the drug repurposing section. And here you can either enter the job ID of a drug or the job ID of a disease. If you enter a disease, you will obtain potential drug candidates that will be ranked by common genes or by common functions. And instead, if you enter the ID of a drug, you will obtain potential indications, again ranked by common genes or by common functions. And this is because we already pre-calculated a list of diseases and a list of drugs. And if you enter your disease or your drug, it will automatically compare your module to this list of pre-calculated indications or drugs. So now I will try to make a quick demo and see if it works. Okay, so if you see, in theory, you should be able to see the webpage of GUILDify, that it looks like this. And it's basically like a search engine. So here you have a text box and you can enter the disease that you want, for example, asthma. Here it already shows you like several options. And you click to, well, you can click to the species that you want, to the tissue that you want, and you click to search. And here instantly it already finds all the disease gene associations to asthma. And they come from different databases. And for example, you can filter by DGenet if you think that the DGenet results are the best. And then you select the score and the algorithm, sorry, which it's not super relevant, but you have different options. And you even have an option to not use an algorithm if you want, just use the seeds. And then you click to GUILDify, and then it will compute the results. It normally takes around between 5 and 15 minutes, something like this, depending on the day. And it gives you a job ID, that this job ID is the one that it will be connected to the results of this execution. And here I already have pre-calculated a results page for one disease that I think it was non-small cell lung cancer or something like this. So here you have the top scoring proteins. You can click here to activate the interactive visualization, which can take some seconds, but it should allow you to check how the proteins are connected. If some of them are disconnected, you can include linkers, which are proteins that connect the unconnected proteins in the module. And you can also include drugs. There are a lot of drugs, so it can be a little bit overwhelming, but then again, you can download the results and then analyze this with the tool that you prefer. Here you also have the functions enriched. You have different types of functions like biological processes from gene ontology, molecular functions from gene ontology, or reactome pathways, for example. You also have the biological functions enriched for the seed genes. And here you have a ranking of potential drugs ranked by how many targets are inside the disease module. So that would be the idea. This is an example of a result for a drug. In this case, it was erlotinib, I think. And then if you want to check the overlap between a drug and a disease, you only have to go here and put the job ID of the disease and the job ID of the drug, or whatever job ID that you want and click to check overlap. And the results page of the overlap looks like this. You have the genetic overlap, which indicates the number of common genes, and the functional overlap, which indicates the number of common functions between the two results. And here you also have like an interactive visualization, which allows you to visualize these common genes, how they are connected in the interactome, which is very cool. And finally, the functions enrich also, and the potential drugs that target this shared mechanism. And finally, like the drug repurposing. So the drug repurposing website looks like this. So here you go to drug repurposing and you click to the ID of the, you put here the job ID of the disease or the job ID of the drug. For example, here I can put the job ID of the disease that I have calculated, which was lung cancer. And the results page looks like this. So for example, here in lung cancer, there are lots of drugs that are related with cancer that are in the top results here for genetic overlap and for functional overlap. It's a little bit different, but they are also related. So that would be it, the demo that I wanted to show you. Now, so to continue, now I will present like a second tool, which is a Python package called NetMed-PI. So this Python package is based on a network medicine pipeline that I learned to use during my postdoc stay in the Barabási Lab. So as input, you need the interactome, the seeds associated to the disease of interest that you want to check, and then the targets associated to all the drugs that you want to test for this disease. And the pipeline is very simple. So first, what you do is to place the seeds in the seeds, which are the disease genes in the interactome. And here, for example, I will show you an example of a study of COVID-19, which was published in 2021 by Daisy Gizy. And they found 332 genes associated with SARS-CoV-2 infection. And then what we do is to calculate the largest connected component, which is abbreviated as LCC, and it means that you select the largest group of connected seeds in the network. In the COVID-19 example, for example, the LCC was made of 208 proteins out of the initial 332. And these 208 proteins are the ones that will define your disease module. So this is much more simple. But now that you have the disease module, we need to calculate the LCC significance, because we want to make sure that the disease module is meaningful. It's not randomly selected. And to calculate this, we need to answer the following question. If we choose 332 proteins randomly in the interactome, would we obtain a connected module of 208 proteins? So to answer this, what we do is to select randomly sets of nodes from the same size and similar degree distribution and check the LCC size. And we repeat this process 1,000 times. And we calculate a distribution of LCC size that looks like this. And if the size of our model is significantly different from the size of the distribution of random modules, it means that the disease module that we have created, it's meaningful. And for example, for this case, we observed that no, it's not randomly chosen, because a random module would have a size of around 188 nodes. And our module is 208, so it's much more larger. And once that we have this disease module, again, what we do is to calculate a ranking of potential drug candidates by using the proximity algorithm that I explained before at the beginning of the presentation. So these would be the main steps that we would need to identify a disease module and obtain a list of drug candidates. And all these algorithms can be found in a new Python package that it's called NetMedPI that was published very recently by Andres Aldana and their colleagues in the bioinformatics journal. And I think that they presented in one of these RePo-SUDOE seminars some time ago. So you might be familiar with it. So in conclusion, using this package and following a similar pipeline would be a good way to obtain detailed results of a network medicine analysis. And the last tool that I wanted to present is a Nextflow pipeline called Disease Module Discovery Pipeline, which we developed recently within the RePo4U initiative, which is an initiative that tries to find drug repurposing candidates and to make like a European platform for drug repurposing. And we made together in an effort with different partners this pipeline that it's very, very useful, but it's also quite complex to use. Why is it complex? Well, because this is an Nextflow pipeline. So an Nextflow pipeline can be a bit more challenging to run, but in this case, it can be executed using a Docker, which is like a virtual machine that has all the programs that you need and all the libraries that you need to execute this pipeline already installed. So you only need to have Docker and provide the pipeline with the interactome that you want, the interaction network, and the seed genes associated with your disease. That's all you need. And then everything runs automatically. So the first step of this process is the module detection, where a battery of 6 module identification methods are executed in parallel. There are 3 different types of methods to identify a module. The ones that connect seeds between themselves, the ones that expand expand from the seeds, like we observed in GUILDify, and the ones that find clusters. And here, for example, we have first neighbors, which adds the nodes that directly interact with the seeds in the module. Then we have diamond, which is basically like an expansion algorithm. We have domino that finds clusters. We have robust and robust bias aware, which connects seeds between themselves using Steiner trees. And finally, we have random walk with restart, which also expands the seeds using a network propagation algorithm until the seeds are connected between themselves. So it's like a mix between expansion and connection. Once that we have the different modules using all these algorithms, we have a next step, which is module evaluation, in which we evaluate all these modules using 4 different methods. One is overrepresentation, in which we calculate the enriched functions. Another is functional coherence, in which we check the coherence of the functions enriched in the module with the ones enriched in the seeds and in other subsets of interest. Then we check for topology measures. So we analyze the module and we check which measures they have. And then we check for how the different modules that we calculated are overlapped, because if the overlap is very high, it already indicates that there is some common ground and that there is something that they are doing well. We also have different types of evaluation apart from focusing on the disease module, which is first to seed permutation, which means that we remove some of the seeds at the beginning, and then after creating the module, we check for the robustness of the modules. And we also check for seed rediscovery, if we can recover these seeds. And then we also have another type of evaluation, which is focused on the network, and it's called network permutation. And basically, we remove some interactions, and we also add fake ones in the network. And then we check if the module is affected by these changes or not. So you already see that this pipeline is much more comprehensive and complex, and we check for a lot of parameters. And finally, for drug repurposing, we use a web server that it's called Drugstone, which basically is a web server in which we can export the disease modules that we obtain from this pipeline. And then from this website, it allows you to calculate 3 different drug repurposing algorithms and analyze the results interactively, which is very useful because it's very, very intuitive. Another very cool thing about Drugstone is that it also allows you to visualize this module and to annotate, to add different annotations. What does it mean to add annotations? It means that you can put the drugs that are targeting the module, side effects of these drugs, diseases that could be related, also include cellular components, which means that if some of these targets are, if some of these proteins are at different cellular components, they are separated. So it's a very cool way to analyze the disease module. And finally, at the end of this pipeline, you also obtain a multi-QC report, which basically it's a report in PDF that summarizes all the execution details and all the obtained results. So that's it. These are the 3 different tools that I wanted to present. And here there are the addresses where you can find them. And here I also leave the email in case that you would like to contact me if you want to use and you have doubts or problems, or if you would like to collaborate with us. We are always open and it would be super cool. So thank you so much for the attention.
Thank you. Thank you very much, Joaquim, for the very interesting presentation about this network medicine and these 3 tools that are very important for the ones that want to study this kind of scientific area. So we now are open for questions. So please, I will ask you to place your questions in the chat. That then we will try to manage the Q&A session.
Okay.
In the meantime, when we are waiting for more questions to arrive, I will start the discussion because I take here some notes, Joaquim. So thank you again. Starting with GUILDify, and so I will put my camera on also so that you can see me. Starting with GUILDify, it exists any tutorial about this tool? Do we need to register to use the tool when submitting a job, since it takes 5 to 15 minutes, if I understood correctly? Can we then recover the results from the job, and how can we export the results? So some technical questions regarding this.
Yeah. So that's good questions. Yeah, actually. So in principle, there are no requirements to use the The web server, the only thing is that it's a website that it's a little bit old, and so when you put the address, you have to put HTTP instead of HTTPS, which normally is put automatically. So if you put HTTPS, you cannot access the server. This should be changed soon, I think, but for the meantime, this is the only way to access the website. But yeah, you don't need to register or anything. You are directly, if you enter the URL, you can directly use the web server. And then, yes, you need to, so you need to wait for the results to compute. But when you are in the waiting area, you are given a job ID. So this job ID, you can use it as much as you want to recover your results. You just have to go here to retrieve results, paste the job ID, and then you will get the results page, or you will be redirected to the waiting area again. So there is no problem in this. It's very easy to use. And yeah, I don't know. Apart from this, I think that's all. How to explore the results. Okay. So if you go to the results page, here you have download options. You can download all the scores, the seed proteins, the interactome. You can download the subnetwork, which is the disease module. You can download the subnetwork with the linkers, which are the proteins that connect the unconnected proteins. And you can also download the drug predictions that are on the bottom. You can, if you go to the functions, you can also download the function. So there is no problem at all, I would say. You can get all the results that you want. Yeah.
Great. Very nice. Very nice. We have one question in the chat already. What are the computational requirements for each of these tools?
The computational requirements. Okay, so for GUILDify, it's a web server, so you wouldn't need any computational requirement. But for the other two tools, so the first one, NetMedPI, is a Python package. So you just need to install the Python package, but in order to use it, you need to know how to program. If not, it would be very difficult to use it. And then for the disease module discovery pipeline, as I said, it's an Xflow pipeline. So in order to run it, you need Docker, which is like a virtual machine that allows you to run something without having to install any libraries or anything. You just need to install this virtual machine, and that's all. But still, you need to have at least some basic knowledge on how to use some basic informatics knowledge if you want to use this.
And do these tools run locally in your computer or in a server? GUILDify, I suspect it is in a server, right? Because it is a web site.
No, locally in your computer, no. Yeah. It's not a website. It's like a program that you have to install, basically. So it would run locally in your computer, yeah.
All of them?
All of them except the website, GUILDify, yeah.
Okay, so thank you. We have another question. What does the score value mean in the table that appears in the ranking of potential drugs section in GUILDify? And which value would you consider as low confidence?
Okay, so yeah, the score of this score this is the functions. So this score here, so it shows you drugs that are ranked based on the score, which is the score that have all the targets related to the drug, sum, but then normalize. So if you normalize it, the one that have the maximum score will have 1, and then the rest will be lower. So that's it. Basically, it's just the score of the targets of the drug.
And do we have any idea about which value would consider as low confidence? So any
Yeah,
threshold.
that's tricky, I would say. I think that it's complicated to know, but for sure you can trust the top results normally. If a value is, so there is no threshold, but the top results normally are quite confident. Honestly, I normally don't use this ranking. I think that this ranking is very useful if you want some initial results or to check which potential drugs could be useful. But then if I want a ranking that it's confident, I would use either the overlap of results, which I don't know. Yeah, either the overlap or the drug repurposing, which is which use the same score. Which here you can check the p-value and the corrected p-value. And with this, you can have a better idea of if a result is confident or not. So I would prefer to do the analysis like this rather than checking the drug ranking in the results web page. But still, I think it's useful. So there is no threshold, but it can give you an idea that it would be quite accurate because the top drugs will always be targeting the disease module directly.
Thank you. Thank you for your answer. We don't have right now any more questions on the chat. This seems that it was a very clear and interesting presentation, I guess. I have another question while we wait for some more in the chat. Nowadays, we have many computational tools for network medicine, for disease networks, for prediction of drug repurposing. In your opinion, how do we know which tool to use, how to select the right tool?
Well, this is complicated. So it's true that there are lots and lots of tools that you can use. I think that a good way to approach this problem is first depending on the data that you have available. If you have data related with, I don't know, gene expression, things like this, try to find tools that could use this input data and fit well the analysis that you want to do. In your case, for example, if this network medicine analysis is super useful for the cases in which you want to analyze a complex disease, you have records of genes that you know that could be related for some reason because you have done a differential expression analysis because you want to start for something and you don't know where but you know that there are some genes that could be associated. So network medicine analysis will always give you a good idea of how to of how this genes are connected between themselves and what options can you use. But it's true that there are lots of tools and it's complicated to choose. Also, depending on your expertise, I would say, again, like we did GUILDify at that moment because it was a web server which was easier to use for people who were not familiar with informatics, like clinician people, people that work in labs. So this is more intuitive, I would say. Even still, the process might be complex at some point, but it's like we try to make it as easy to use. But then, for example, if you already want something that it's more detailed, you have experience with bioinformatics, or you have people who could do this analysis, I would go for more complex tools like the Nextflow pipeline that we presented, which then it gives you like a more comprehensive analysis of potential disease modules. It evaluates it in many different ways. And in the end, you end up having a lot of results that might be much more meaningful. I don't know if I answered the question correctly, but it was complicated, I guess.
It was actually an open question, so thank you very much for your input. Actually, these tools are very, very important for the determination of the disease targets and the predictions of drug repurposing. That is also our goal here in the RePo-SUDOE project. It seems that we don't have more questions in the chat. So with this, I think we can close this webinar, this another RePo webinar, and thank you, Joaquim, again for your availability to share this presentation with us, and we hope that this will be very useful also for the scientific community to pursue their studies on drug repurposing. So thank you very much.
Thanks to you for inviting me. And if anybody wants to use these tools, feel free to contact me and I can try to help.
With some comments, it was really well explained. Thank you. And so indeed, thank you very much. See you on the next RePo webinar. We will announce it sooner for next month. Thank you. Bye-bye.
Bye-bye.